online. So, good afternoon, everyone. Thank you for joining us in our symposium. It's an honor and a pleasure to be here in front of you as one of the chairs of this event. So before we start, I need to again thank Dr. Skell for her hard work, for her vision in bringing together all these professors, researchers to be here with us today, sharing their knowledge. And it's very important for us, for our students, for our program. So thank you very much, Skell. If it wasn't for you, I will say it again, that will not be here today. Thank you. Also, I want to thank our dean, the post-graduation programs, the oral biology and the oral rehabilitation programs for all support that they gave us. And of course, I would like to thank our IT guys. Paula was not here, but probably they were listening to us for all the technical support. Because of them, we can transmit this, broadcast this event online. So today we are privileged to hear from an outstanding group of speakers who will share their knowledge, their insights, and the latest research on advancing biomaterials from research to multifunction innovation. And as speakers, we have Professor Owen Addison from King's College London, who will guide us in studying natural and synthetic biomaterials in a different light. Professor Valentim Barão from FOP Unicamp, presenting on biomaterials applied to implant dentistry, a multidisciplinary approach to the development of new strategies with antimicrobial potential. And for the last but not the least, Professor Paula Ramos from Chemistry Institute. I cannot say it in English the acronym of your university, I'm sorry, and sharing insights on the nature is an inspiration for the synthesis of functional biomaterials. So as I said this morning, I encourage all of you to actively participate, ask questions, and don't miss this opportunity to first, make new friends, because I think we are here to have fun always. And second, to make new collaboration, to new partners, and to improve your communication skills. That's why we're speaking in English here. Not just because we have an English professor, but I think it's very important for us to try to communicate in another language, which in this case is English, because it's the universal language of science, okay? So once again, thank you for being here. I hope we will have a wonderful time together and enjoy this symposium. Professor Owen, the stage is yours. Thank you.
Thank you very much for that very kind introduction. Can everybody hear me okay at the back? Good, okay. Let's try and work the IT here. So I thought I'd just give you a little bit of an introduction to who I am and where I'm from, first of all. And then I'm going to explain why I'm in Brazil this week and the last week. And then I'm going to talk you really through a journey of my own science, and in particular talk to you about some techniques which may not be that familiar to you, but hopefully will excite you. So this is where I'm based. I have probably the best view in London for a dental faculty. Our labs look over Tower Bridge, and we're very fortunate in that regard. We're a huge dental school for UK. We're the biggest dental education providers in Europe. We have over 1,000 undergraduate students. We have over 250 postgraduate students. And we provide specialist training in every single discipline. I lead a different center within the faculty. So we're about 2/3 the size of the whole faculty of dentistry. We cover most of the clinical areas, and we have particular expertise in areas like biomaterials, devices and diagnostics, artificial intelligence, and so forth. One bit of information I pulled out just a couple of days ago, though, was this figure on the right-hand side that shows our research collaborations over the last 5 years. We're a really internationally looking faculty. We want to collaborate with the best scientists across the world. And I was very glad to see that one of the biggest circles that we see here on this graph is Brazil. So we are invested in working with colleagues in Brazil, and my colleagues I know have lots of collaborations going forwards. But why am I here? Well, I've been coming to Brazil for nearly, I guess, about 12, 13 years now. I started coming to Brazil when I was at the University of Birmingham. I was charged with trying to create some networks and some new opportunities. And I've been hosting students through CAPS, through FFCLRP schemes back in my own lab over the last decade. And I've got to know some really outstanding scientists. But the other reason I'm here at this time, and this was last week, is the building behind where my friends and colleagues are standing. I have been involved in using a set of experimental techniques that we've developed really strong pipelines for over the last 15 years. And I'm super excited to say that I've now done my first research experiments using these facilities in Brazil. So this is what I'm going to explain to you what I do. So I'm going to go through something called synchrotron resources or sources. I'm going to describe some opportunities and approaches. And then I'm going to talk to you about some case examples. And I didn't know what my audience was going to be today, so I thought, well, bone, teeth, polymers, implants, let's cover everything. So I'm going to talk to you about specific case examples using these sets of techniques. But let's start off with what a synchrotron is. So occasionally, governments decide to spend hundreds of millions of dollars to build something called large experimental systems. The biggest showcase is the one on the left, which is built underneath Switzerland, which you might have heard as the Large Hadron Collider. But there are other sources, such as the ones in the middle, which is the Advanced Photon Source in Chicago, which is an X-ray or synchrotron light source. And then we have particle sources, which is such as ELL in Grenoble in France, which is a neutron source. I'm going to focus on the middle one, although I do use facilities such as neutron as well. So what is the synchrotron? Well, top left, you're looking at this big building. This is Diamond in Oxford. And in the right-hand side, you're looking at the inside of the Swiss light source in Villingen. And what they are is effectively a supermicroscope. They are developed to produce light that is so intense in flux that we can do measurements that are just not possible with any other form of laboratory technique. They give you light in the whole spectral range, from visible to hard X-rays, and they also allow us to manipulate those X-rays in a way that you just cannot do in your own laboratories. And they're accessible to dentists. I'm a dentist by training. I still do 50% of my week on clinics with patients, and I've conducted over 50 separate experiments on 3 continents doing this type of work. Come in. So what is a synchrotron light source, or how does it work? I'm just going to briefly try to explain how this facility works. What we have inside of these donuts is an electron gun, which is number 1. The electrons fire into a small accelerator, which is number 2, and then they're released into the big ring, which goes around the outside and accelerates these electrons just below the speed of light. So they're going incredibly fast. And there's a phenomenon that happens with electrons, which is when you move them around corners, you accelerate or deaccelerate them. And when you do that, you release light in the form of photons. And what we've learned to do is capture that light. Let's come back. We've learned to capture that light and take the light off on these tangents and start to use it in individual experimental areas. So this is Sirius down here. Sirius is in Campinas. It is a beautiful new facility, and it is at the moment occupying about 16 instruments, but they have the capacity to build up to 40. And one of the remarkable things about this type of facility is you can access it for free. And that's incredible, because if you had to to go and buy some top-end laboratory equipment. You know, you're spending years writing grant proposals to try and get it to happen, and then you have to maintain it. Here you can go to something that is world-leading to conduct your measurements. So why is this interesting? Well, you can do huge amounts of things with this. You can work on tissues, you can work on materials, you can work on live cells, you can work in hydrated conditions, you can work on living conditions. You can work from angstroms down to up to micrometers, up to millimeters. You can work incredibly fast, and it allows us to do, you know, a really wide range of imaging techniques. And this is what the inside of one of these looks like. This is an end station where we try to conduct our experiments. But one of the things I would point out here is we build this setup next to the sample to do whatever methodology that we really want to. So if I want to do diffraction imaging, or if I want to do chemical spectroscopy, or if I want to do tomography, we can manipulate that end station to work for our samples. And so these are a range of techniques that I'm not going to go into, but the primary techniques that I work on, and I'm going to go into them in a little bit more detail when I show you the case examples. So I'm going to run you through the last, I don't know, 4 or 5 years of work that we've done focusing on these areas. So we're going to start off by talking about bone and a different hypothesis that I'm challenging at the moment that is only possible because of the way that we're imaging. We're going to move on to some really new work that we've just published, trying to study the structure of enamel. We'll talk a little bit about photopolymerization as a completely different challenge. I'm going to talk a little bit about implants, and then if I have time, I'll go to my pet subject, which is, let's finish on dinosaurs because it sounds like we can wake you up. So let's go. And please, you know, if you're really confused, wave a hand and I'll stop, because I've been told that I speak too quickly. And so I'm going to try and slow down over this next piece. So the hypothesis that I'm going to challenge you with, because I've seen some fantastic research this morning about bone augmentation, is what is grafted bone? I look at this X-ray, and this is an implant that is 7 years of success. The site was grafted ahead of that implant placement, but this is at 7 years. Who here thinks that looks successful? I think pretty well all of us, because by all of our definitions, we have bone above the most superior thread, and everything looks, you know, particularly good. But we know that survival of implants in grafted bone is worse than when we place it into native bone, and there are reasons that we understand for that. And now I'm going to show you this. This is the same implant at 10 years. So between 7 years and 10 years, I'm going to go back, between 7 years and 10 years, this has happened. Now, this is obviously very severe periimplantitis, but it looks odd. It looks very much like we've only lost bone where we've grafted. And so I've started challenging myself that grafted bone is not grafted bone in the sense of de novo bone that works and can maintain itself in the same way as healthy bone. So, how do we image? Well, we often do things like you do it here. We take an animal study, maybe a calvarial study, we'll do some micro-CT. Micro-CT gives us this sort of resolution as we go down. So this is the sort of the typical quality of micro-CT that we can do. What we're looking at here is different materials placed into these defects, and we can sort of tell the difference between where the graft material is and the native bone, and we can pull out some of the measures that I've seen so nicely presented this morning. But is this enough? Are we missing something? So this is the same image, or the same tissue image, firstly with a lab CT at 5 microns resolution. On the right, I'm going to show you world-leading resolution of tomography. So this is now at 40 nanometers of resolution. And suddenly we can start to visualize the crypts of osteocytes and the canalicular network. So what do we do with this information? Well, to get there, first of all, I should explain, we have to use a synchrotron source. You cannot do this with a lab source. And there are different ways that we get there, but the way that I approach this is through a unique technique that's based in France at the moment, the European synchrotron, which is a technique called holotomography, where we fire light through the object, but instead of measuring how much light is attenuated, absorbed, we measure the shift in phase. So this is one of the things that is very different about the light produced in these facilities, is that we can also control how coherent it is, so how much in phase it is. And by working on these samples in this way, it allows us to do what we're going to do next. So one of the things that is a difficulty with this type of measurement is we have to really understand how to make samples that work. We have to worry about beam damage. We have to work out how to align in a very, very precise way. So it is something that is a learning experience to get to. But I'm going to show you now, hopefully, the journey that we've been on here. So my hypothesis is that bone doesn't work, a grafted bone often does not work as bone because it is a composite of the graft material and the newly formed bone with some fibrous material in between. And one of the cells we're particularly interested in is the osteocyte. The osteocytes are the most common cells within living bone, and they form a network that provides its mechanosensory control and its control over its mineral density. And so one of the things that we set out to understand is, if we can image osteocytes at this resolution, does that osteocyte network, does it reproduce from different types of bone to different types of bone? And then does it reproduce into grafts? So this piece of work, we coined the term osteocyte connectomics. This is it's a 2D projection of the type of imaging data that we provide. And what we're interested in is how these osteocytes are connected to each other within this structure. On the right-hand side, these are now the types of variable that we can pull out of this type of imaging data. So for those of you who presented beautifully this morning, you have 5 or 6 common parameters that you can pull out. Once you go to this resolution, you're now into a larger list, which may give you some particular good insights. So what we started doing is working on jawbones, femurs. We also started working on pathological bones. There's something called heterotopic ossification, which is bone that forms in soft tissue sites very, very quickly. And we wanted to understand how individual osteocytes are talking to each other. And by doing that, we found out that there are fundamentally only 4 or 5 ways that they connect. And we found out that that connection was very, very robust. So we started doing some simulations to see how fluid would flow between osteocytes using a very simple form of something called computational fluid dynamics. And so here, what we're doing is modeling 2 cells talking to each other or connected to each other, and we're modeling what would happen if we disrupt the connections between them. And what we found is everywhere we looked, you had this over-engineered complex, which means that if you did disconnect, 30, 40% of the canaliculi, there was still a way that the entire body of osteocytes were still connected to each other. And this seems to be quite a phenomenally, clever thing for us to have evolved to be able to do. And then we come to graphs. And so here's the same type of imaging data on the right-hand side. What we have here is just a projection through that tomograph with the graft material in the middle that hasn't completely healed. And then we can see the osteocytes are starting to form around that grafted material. And hopefully it projects well enough in here that we can see that actually where I've drawn the circles, the 2 red circles, in between there, the mineral density is higher. So what we've started to understand is that unless you reform this osteocyte network within a graft, the body is no longer under control of its mineral density in how it would normally do it in health. And this is why I believe that the rate of bone loss in periimplantitis is very different to the rate of bone loss in a non-grafted site. Okay. Let's go to case 2. So case 1 is all about imaging, a high resolution. Case 2 is about using chemical imaging. So something we call spectroscopy. You've probably come across it in terms of vibrational spectroscopy, so FTIR or Raman. And we set ourselves this question: can we image the crystal-like orientations of that flat tooth surface? So a surface technique without doing really any preparation to the tooth whatsoever. So the idea here is that we don't want to make thin samples, we don't want to do embedding, we don't want to slice the tooth. So we took the case example of dental erosion. My clinical teams are very, very big into treating tooth wear at King's, and we understand some of the parameters that lead us to having progressive enamel loss. And what we know is that we can normally detect this using things like profilometry, where we measure how much of the surface gets removed. We can measure it with indentation, whether that's micro or nano. And we can measure in slices through the teeth the structure of the tooth. However, I wanted to do this with no sample preparation whatsoever. So we looked to a technique that's used in geology at synchrotrons. We didn't invent this technique, we just decided to apply it to a different area. And one of the key things about the light that's produced at these types of sources is that not only is it very, very bright, but we can control something called its polarization. So if you remember that light is both a wave and a particle, the orientation of that wave gives us its polarization. So the technique is a chemical technique, it's not an imaging technique. We fire X-rays of a specific energy, and if they're absorbed by an atom, we can knock out one of the outer, we can knock out one of the orbital shells. So one of the electrons will be knocked out, and then another will jump into its orbital shell and release a photon, which we can measure. So from this technique, we can normally just say, oh, this is calcium, or this is phosphorus. But by doing something a little bit more with it, we can actually start to say, well, this is calcium bound to oxygen, or this is calcium with its nearest neighbor being hydrogen or something like that. So for this technique, we identified that there was an interesting relationship between the direction that we took our polarized light, so either straight to the crystals in that direction, or crystals in that direction, with a very subtle change in the way that the calcium would be projected in this spectroscopy technique. I'm trying to keep it as simple as I can in this one. And so we exploited that by saying, well, if we start to image the tooth surface in different ways, knowing that we're changing our polarization, we can start to say, well, those pixels are orientated this way, these pixels are orientated this way. But we run into a problem. Because if you look carefully at the graph at the bottom, there's only 2 options. And we want to have a third option. So to get around that third option, we rotate the sample, and then we can identify the third piece of orientation. So the work that we've done here for the first time is to map the surfaces at 2 energies associated with the calcium absorption edge, and then by rotating the surface, we can start to develop an understanding of crystal structure. So very, very simply, we have something mounted on that little blue insert there. That's a rotational stage. The bit with the yellow that you can see is one of our silicon drift detectors. And effectively, we're going to fire X-rays at that surface, and then we're going to map the surface and then rotate. For the eagle-eyed amongst you, you can see that I've added some squares onto here. We lasered some fiducials to allow us to be accurate in our repositioning, although that was a bit of just being over careful. So this is the concept. Depending on where the crystal is orientated, it absorbs energy in a slightly different way. And by rotating through, either rotating the beam or rotating the sample, we can start to work out these orientations. One of the things that's really important is we can also work out, like killed your computer here. Let's try this again. Oh, let's go back a few. Okay, one of the other things that we can do is we can work out the pixels that aren't moving, which gives us our ones that are end on. And remember, enamel principally has our crystallites coming end on. So here's a very simple experiment. We work with a cross-section to prove that this actually works, and then we work on buccal samples that are completely unprepared. And this is the type of data that we'll end up generating. So we'll end up generating an absorption energy for each of those different angles, and by looking at the peaks, which are going to be separated by 90 degrees, we can then work out an orientation. And from that orientation, then we can work out which ones are also end on, because they have a lower energy difference. And then from this, we can start to create these textural maps. And I'll just now show you what these maps will look like. It will give us our full orientation, but also tell us how ordered the sample is. So if everything is really well ordered together, it means we have high texture. And if everything's a bit more random, we have low texture. So what we have done, and we published this just at the beginning of the year, is created a new approach in dentistry to study surfaces with almost no preparation whatsoever, that allows us to quantify, not just visualize with SEM, but quantify crystallite orientation and crystallite texture. And we use that in erosion studies to understand which populations of crystals within dietary erosion, or say stomach intrinsic based erosion, are actually the ones that are being most affected. And what we've worked out is that is actually those that are our low angle population seem to be preferentially removed, weakening the interprismatic structure. So I put a couple of pictures of a few people in as we go, because this is actually pretty complicated on paper, and I'm sure not really necessary. Many of you would have followed me talking about that in a very quick way. But this is, say, Lynn, who's next to me here, is a dentist with no physics-based training, no coding training whatsoever at the outset of this PhD. She's now just finished year 2. Her first paper that she's ever written went straight into Acta Biomaterialia, and she's done all this work. So you can, the attainment step to get up to working with these techniques is really achievable. She is exceptional as well, but that being said, I've had very, very good experience in my juniors coming through. I'm glad Anna Paula's here, because Anna Paula said something quite important earlier on, which she said we were talking about chemical processes, and we're talking about biological processes which have a chemical basis in terms of healing. And she made the statement, just because something is quicker doesn't mean that you end up with a better material result. So I'm going to take you back to something, I'm a dentist. You can see all of my problems that I'm following are just normal dental questions that we answer as researchers. So, very simple question. How quickly should you cure a photopolymer? All of us who are dentally trained to place white fillings, we take that blue light, stick it on above our tooth, press go, and depending on the irradiance that it outputs, depends on how quick we get to initially a gel point and then to a full maximum conversion. Companies want to do this in 3 seconds because they think we as dentists have got short attention spans and want to do everything super efficiently to save a few minutes. Personally, I'd never thought it was a good idea. And I'm absolutely certain that Anna Paula's now going to sit there as the chemist in the room and say, well, she knows it's not a good idea, but we're just going to prove what she already knew. So what we did, working with one of my other absolute stars within the group, this is Dr. Slobodan Sirovica, who's now based at Queen Mary, we decided, well, let's just build a way to photocure in a synchrotron. So when I went back to that original slide at the beginning where I showed this massive space where we can build stuff, well, we thought, well, let's go and do this. Let's build a light source within the light source, and let's combine it with some other techniques that allows us to look at the structural evolution of a composite. We did it a little bit more complicated because not only did we want to measure the structure of the polymer, but we also wanted to measure its conversion. So we also put a spectrometer in there. So we did some near-infrared spectrometry at the same time. And then we have a light guide to cure. So we had effectively a dynamic measurement of structure, simultaneous with conversion, and with high control of irradiance. And then it produces something as boring as this. This is sort of the output of our X-ray scattering. For those of you not familiar with the terminology we use, this is what we call the Q space, which is measured in inverse angstroms, and this is our intensity here. And we started off, and we started with our monomers, which would be the dotted line, sorry, the closed line, and then we cured, and 6 seconds later we're at the dotted line. And you think, well, what can I really do with that? Because it's just showing I've gone from one material to the other. But what you can then do is actually you can start to solve it. So you start to take other components that you know are in that monomer system and you fit them against these broad peak features. So over here, we're doing some least square mean fitting to try and work out how we solve what this scattering feature is actually giving us. And this is what we find. So this is just the summary of what we understood. And what we found is that if we ramp up the polymerization rate, which is down here on this axis, and we look at how initially the monomer extends to become a polymer, depending on the rate of cure, and what we're looking at in the different colors are systems that have different viscosities, but also we have different photoinitiators, we end up with a difference in initial strain, and then also we end up with on the second one here again polymerization rate here against how ordered that structure is. And again we see if you go very fast you end up with a more disordered structure compared with if you go very slow. Anna Pardo, I'm sure, would have predicted that result, but it's nice to be able to actually show it empirically. However, that wasn't a composite. So we've demonstrated to ourselves that photocuring very quickly is probably not a good idea in the polymer, but let's try and do it with a real composite. And to do that, I can't do it with X-rays, because the problem is all those filler particles get in the way. They scatter the light in all different directions. But what I was very clear about at the beginning is that a synchrotron produces light across the spectrum. So it also produces visible light, and it also produces infrared. So now we have super infrared. It's actually not that much of an advantage over a lab source. It's about twice the flux, but it's slightly more structured. And so now what we're trying to do is see if we can detect the same things, strain and conversion, in a completely different way to anybody's done it in dentistry before. And so this is what we managed to produce. We managed to look at a composite at a single particle level. We managed to map degree of conversion with respect to the particle centers. And because we've got such high quality spectral data, it also allows us to look at very subtle shifts in that data in order to pull out something we call molecular strain. So we can now understand that, again, if you ramp up your polymerization rate, how well you're going to convert at an interparticulate level, but also from how much strain you generate in the system. Again, on the left, I'm not going to go into huge details of what we're showing here, but effectively what happens is you have a boundary layer around the particles that has lower conversion and lower strain, but it is set by the composition and how quickly you set the polymer. On the right-hand side is a different type of measurement that the reviewers asked us to do, which is something that you don't do it necessarily at synchrotron, although you can, which is now called indirect FTIR spectroscopy. And it gets across something that we know as the diffraction limit in infrared spectroscopy by using a different form where you take an AFM tip, you put the AFM tip on the polymer, you shine mid-infrared light to the base of the AFM tip, and then the surface slightly moves because it's expanding from absorption, and you measure, you can generate spectra indirectly. And we managed to demonstrate exactly the same effect on a single particle. So that was the first work I ever did on a composite, and it went into Nature Comms in end of 2019, beginning of 2020. All made because of access to a different way of measuring. Case 4, how am I doing for time? I'm doing okay. So this one, I don't know if anybody was sort of taking photos, but not to take any images of this case because it's unpublished. This has been a piece of work that I've been doing for nearly 10 years now, and I've just got to the point where I'm probably happy with it to put it out. So the simple question is, do titanium implants corrode in the body? It's a hypothesis that actually I was one of the very first to propose back in, I don't know, it was 27, 26. I was very uncomfortable with the idea, particularly when the BBC phoned me up from the original paper that we put out, which was just small print in RCS Advances, RCS Interdisciplinary Science. And they said, you know, should we be telling patients not, to have their titanium implants taken out? And I said, absolutely not. This is mechanistically a concept idea, but it could have some value. Unfortunately, a lot of people around the world, particularly in Germany, have taken the idea as a research idea and are sprinkling titanium on cells and putting titanium chloride into cell culture, which makes absolutely no sense whatsoever, to try and drive this hypothesis that titanium is a materials determinant of implant health. But what we do know is materials can cause inflammation. So this is a classic case of what we call metal-on-metal implants. It's based in the orthopedic sector. And what we have here is a total joint replacement with a femoral stem, and then into the acetabulate cup. And what they found out was that if you didn't put a bearing in here and you went straight on metal on metal, here we're using cobalt chrome molybdenum, you generated conditions where not only did you have wear particles, but you're also producing a lot of solution species. And back in the day, and I'm not going to go into the mechanisms there, but back in the day I was asked to work with the orthopedic surgeons to see if they could understand why they were getting different histological results associated with some of their explants. And what they found was that they were, when they were looking into histology, they were seeing cases that were either sort of very diffuse cellular infiltrates, plasma cells, lymphocytes, but in certain cases they started to get more organization. In other cases, they started to look something like what we call a tertiary lymphoid organ. And a tertiary lymphoid organ is effectively a collection of T cells, B cells, and it has to form in reaction to recognizing something. It's not forming because it's just come across an ion or a particle. It's forming because it's either came across a protein that's misfolded, and we call it like a hapten-like response. And so what we did for that team was to take some tissue, initially working at quite low resolution at the top, but then starting to map some of these tertiary lymphoid organs and then overlay metallic responses. Again, using a synchrotron, using something that allows us to get a probe down to 1 or 2 microns in size and allows us to look at very, very dilute concentrations of metal. And we demonstrated that you had this high correlation, particularly with the chromium signal, which was being ignored at the time, with our TB cell infiltrate. And that was sort of, that's over a decade ago. But of course it points to the people who are turning around and saying, well, does titanium corrode in a dental situation? Well, let's look at the fundamentals of this first off. The titanium oxide surface layer should not spontaneously depassivate in physiological conditions until you get, at the earliest, up to about 68 degrees centigrade. So it is highly corrosion resistant and should remain highly corrosion resistant if we just think about a neutral physiological environment. But there were a couple of people at the time just throwing a few things out. And one of the most important ones was from the Montes Bayan group in Oviedo that did some beautiful mass spectrometry and demonstrated that in patients that had titanium prostheses, they had titanium bound to transferrin, which is your main chelator, or main sort of carrier of iron. It keeps iron bound in extracellular fluids and in blood. They found titanium had displaced it, which means that at some point titanium had to get into ionic solution in order to end up on a protein. It's not a particle. So we decided to follow this story, and this is something, again, we've been working on for years because it's taken a long time to get the biological replicates, but we would take tissue from the patients, do the histology, and then go to the synchrotron to try and understand what we have. And so the data I'm showing you has never been published. It is sort of written up and ready to go now. It forms the basis of about 25 individual inflammation cases, and over, I forget my exact number, but we have taken somewhere in the region of 100,000 individual spectroscopy measurements to understand titanium chemistry. So what can we do? Well, we can start off at low resolution of the tissue imaging, which you can see here. Everything in red is titanium. You'll actually see that we can see the rete pegs of the epithelium with the other signals, which are phosphorus and calcium. So we can see that we're forming or finding titanium at random in histological sections. And then we go to higher resolution on the right-hand side. Again, we see it all subepithelial, and we're starting to see something to do with the size. We go down even to further resolutions using different instruments. We can see, sometimes we see sort of granulomular type formations that we've correlated with histology. But the key thing is when we go again to the sort of the highest resolution that we can, here this is at 50 nanometers, we see individual pixels. So we have nanoparticulate sizes of titanium around an implant. Does it matter? Have no idea whatsoever at this stage, because it's just there. Just because something's there does not mean that it's important. So next question, how much of it's there? And so one of my PhD students who did their PhD at the synchrotron started working on quantitative techniques to try and work out how many particles would be estimated in a small area. And the numbers are actually quite remarkable. So this is really nice quantitative work. I put a TEM up here to show sort of what an individual particle would look like. But what we found is that in different tissue sections, so this is in a millimeter cubed, you would have something like 1.5 times 10 to the 5, 3.7 times 10 to 6. So we're talking like approximating like 4 million particles within a 1 millimeter cubed piece of tissue, which doesn't sound too good in terms of a biological response. So by doing this, and again, I said we've looked at nearly 25 individual patients with about 50, 60 specimens interrogated over this time. We've been able to understand what the mass fraction of titanium is, the size of the titanium, but the other thing that we should also be looking at is what chemical form is that material in. So that takes us to where we use another technique, which is called X-ray absorption spectroscopy. We covered it a little bit with the tooth study, where we can start to get fingerprints for individual metallic elements here. And what we tend to do is we tend to work against standards, which we know exactly what those materials are. We can compare what we find from the tissues, and then we start to develop an understanding. And what we find is that you have highly mixed species, but these spectra have high chemical variability at submicron levels, but they principally form into two types. So they're either crystalline oxides or they're amorphous mixed species. But that doesn't necessarily help us that far in this story, so we have to go even further. And we say to ourselves, well, can we prove that you could generate those in physiological fluids in a way that would potentially happen in the body? So again, one of the remarkable things about the instruments that I'm using is I can go from one day mapping tissues to the next day having an electrochemical experiment where I'm doing in situ dissolution of titanium in biological fluids. And by modifying those biological fluids, we then built up a picture that we could explain if we put in things like small amounts of reactive oxygen species, where you've, small amounts, say, of transient peroxide, that you could actually start to produce these. So under pseudo-inflammatory conditions, you can account for titanium in solution for a very short period of time. And we've taken this work further to try and understand what environmental conditions, what biomolecular content would be required in order to completely map onto what we see in the tissues. And so hopefully we'll submit this within the next month or so, but it's a huge piece of work. I mean, I'm not joking, it's about 10 years of work. And we've been very cautious about the message. Do I think this is biologically relevant? I still don't know. But it's important, at least for the people who are trying to answer that question, that they understand what they should be stimulating and working towards. The other thing that straight jumps to mind for all of those of you thinking I'm going to jump to zirconia implants is we've been doing some work on zirconia implants as well. And guess what? You find a load of ceramic debris, also on a very small scale. And so there's now a question of whether you're getting sort of very minor low temperature degradation of that surface, leading to sort of degranulation of the ceramic. Okay, so I've talked to you about high resolution imaging. We apply it to bone. I've talked to you about structural analyses we can apply for teeth. I think I've talked to you about dynamic measurements, understanding polymers, and I've shown you about using chemical measurements to understand chemistry within tissues. And so I'm going to end it on something that's far more interesting than that, and that's dinosaurs and Komodo dragons. So this is possibly the most fun I've had in a project in a long time. Just before lockdown happened in the UK, a Canadian colleague came across to us on a Marie Curie Fellowship, which is a European scheme. He brought his family. He arrived, masks on, we sat in a room, and he had this whole plan to do this other project. And we were like, well, we can't do any of it, because he was having to go and do visits to other units around Europe. And this guy, Aaron LeBlanc, fantastic Canadian developmental biologist and paleontologist by background, sat with me and we said, well, what can we do with what we got? And he turned around and said, well, I've got these tyrannosaur teeth, these fossils of some tyrannosaur teeth, and they've got some really weird enamel. So we as really arrogant mammals think that our enamel is something really special. And we tend to ignore simpler forms of enamel because, well, they're not as interesting. They're not, they don't have prismatic structures. They look really disorganized. But then we sat there and we said to ourselves, well, these giant things, like including the tyrannosaurs, produced enamel that just doesn't make any sense. It was only like 500 microns thick, non-prismatic, completely disordered. But they grew it into things that looked like a steak knife, that they would run up and down the front and the back of the teeth. So you have these serrations made out of enamel on these. So they were obviously important. So they created this important structure out of something that is considered to be much more boring in terms of enamel development. But the other thing that I, as the dentist, was thinking was, well, we know that prismatic enamel is produced under high control of your ameloblasts, and we're trying to reproduce that as dentists in regenerative dentistry. But we know that these reptilians are producing it with the cells much, much further away from the mineralizing front. So it's much more under chemical control than cellular imprinting of the structure. And so the hypothesis flips to say, well, can we learn something about regen of our enamel if it's more chemically focused rather than under cellular control by looking at other things? So that's how we started looking at tyrannosaurs. But the problem is tyrannosaurs' teeth are fossils, so really they're rocks. So there's not a huge amount that you can tell apart from with the enamel. The dentine itself is something different. So we had to find something that looked the same, which is where these magnificent things come into play. So this is a Komodo dragon. This is one in the Singapore Zoo that I took a picture of the other day. We were very fortunate during lockdown because we had this new hypothesis, and I turned around to my guy and I said, wouldn't it be great if we could get a Komodo dragon tooth, because we know just the same they've got these serrations. And he turned around to me and he said, you're an idiot, because these are the most protected reptiles on the planet. You can't just get one. And a day later, the Komodo dragon in the London Zoo died. And we got a Komodo dragon. And so we started applying this whole suite of techniques that I've shown you before to these structures, to tyrannosaur fossils, and also to these incredible teeth, both erupted and unerupted, from Varanus komodoensis. And what I'm showing you here first is if you just look under our conventional techniques, SEM, even TEM, you can miss things. And so these are images on the right-hand side of crystallite orientation within these incredible serrated structures. And there's two things that you can see, and probably not projecting very well, I'm afraid, but I've got individual lines here that point to the direction of these crystallites, and then I've also got in the heat map texture where it's more orientated in the same direction the brighter red we go. And what we found is that even though this is boring, reptilian, unprismatic, not a mammal, an ammal, they were weaving their prisms. Sorry, they were weaving their crystallites not into prismatic form to give, I assume, a mechanical advantage. And then we go to Komodo, and we found, well, we were ready for our fancy paper saying that we've got the solution to this. And then something rather unexpected happened. Because even though they looked the same, they were doing something completely different. So these are a different type of measurements at the synchrotron. These are elemental maps, where orange here is iron. And what Komodo's done, and it's evolved in a completely different direction, it's not trying to structure its enamel from a crystallite perspective. It's iron-tipped. It's cutting edge. And we can even go down as far as saying the chemical speciation of that iron edge. And so that was, as I said, a fun thing to do with the suite of techniques we had, but actually has answered quite an interesting question to us. And now we're really focused on reptile enamel in terms of how we can actually reproduce some of these structures purely through a chemical synthesis perspective. So I'm really sorry that it would have been very quick to go through lots of different types of ways that I'm using these types of measurement. We are using them to answer old questions, just in a different way. And I think by doing that, we are getting completely different insight to what's gone before. These facilities have been built for you to access. So I was very, very fortunate to put a proposal together with our champion host here today with Hiskel. Hiskel was with me in London for 6 months. We managed to do just a little bit of visitation to the UK resource when she was there, but we made this plan to do some measurements here at Sirius. Sirius is a fantastic instrument. It really, really is, and it's full of super motivated young scientists who are really trying to make your measurements work. And it's free if you get your proposals accepted. So you write a small grant, It's 2 pages. They look at your feasibility of your measurements, and if they think they're feasible, it goes to the next stage, and then a small science committee will look at it and they'll rank them in terms of what's more useful to use that time. And when you go to them, they work you very hard because you're expected to use the time that you have. There are a few things about it that take some time, particularly the data analysis. But I've always found that every new technique I've tried, there's somebody who's there who's willing to help. And now I'm the person willing to help in the other direction. So I've got a fantastic team who all do different aspects of the types of data analysis we have. Been lucky enough to be pretty well funded on this type of thing. And I put up my papers from the last 4 years using these just to see, where we're managing to land them in terms of, I don't want to chase high impact journals, but at the same time, if you put them into these types of review process, you get absolutely clobbered in terms, and they force you to really make sure that you are saying something that you can really back up. So my team's there, I think, has expanded slightly since then with Eleni and Hiskel and both now done experiments with me, and obviously their collaborators and their PIs working with them. So that's me for 45 minutes. Happy to take any questions.
Congratulations, Professor Owen, for your incredible talk. If someone want to ask some question?
Far away.
Congratulations for your talk.
Thank you.
You were talking about small particles, titanium particles. Have you observed that they were endocytosed by macrophage and giant cells, or in the extracellular milieu?
So the part that I've not shown here is the histology that we do along the side. And you do really see a mixed inflammatory events here. You certainly see things that look sort of granuloma-like with sort of foreign body reactions, but you also see sort of mixed lymphocytic, very sort of diffuse
in the extracellular matrix.
Yeah, absolutely. And then we've done a huge amount of in vitro modeling, where we've looked at neutrophil reactivity, macrophage reactivity, macrophage polarization, stromal cell responses. We've even gone as far as asking the question, does it get modified once it's out into the environment? So again, we're interested in the chemistry, and we're saying to ourselves, well, we've done experiments where we cycle it through macrophages, so allow phagocytosis or even endocytosis, something, lyse those cells, put the lysate back into another cycle, and keep measuring chemistry. And it really, we don't think there is any cellular mechanism, despite a phagosome having quite aggressive chemistry of changing these particles. So we think the particle speciation is set as it comes out into tissue. As a separate study, I was called in by a company who had a problem with a new surface, and we found the problem quite quickly, which was that they were one of these companies that were using laser etching to create a topography, and they'd lost their atmosphere during this. So instead of doing that in an argon atmosphere to prevent oxidation, they'd had a problem, and they sent this batch of implants out where the surface had been grown thermally from being tens of nanometers of oxide to like 2-3 microns of oxide, and you could see it under the SEMs just delaminating. And we had the failed implants and the granulation tissue from patients. And the remarkable thing was, I mean, some of it was, you know, it was like taking, I mean, it was just ridiculous, the amount of metal. But the point was, their failure rate only went from 3% to 11%. So you have to assume that there was 89% of the patients that had all of this coming off during the healing phase, and it made no difference to them whatsoever. So I think it's, you know, you would have seen in the literature this idea of, you know, and there's these, I'm sorry, there are absolute terrible reviews out there of because they miss the underpinning corrosion science that goes behind this, of what happens with metals in tissues from dental implants. I would take most of it with a real pinch of salt. But do I think it could subtly modulate responses in certain patients? Probably.
Okay.
Probably, yeah. But I'm not going to sign my career away on that message.
But you think it still has to be demonstrated, right?
I think you have to somehow link levels and speciation with failure. And I mean, so I'll give you an example of one of the huge problems that we have, is that you know, there's a load of studies that are out there looking at dental implants and titanium release. And well, why is toothpaste white? Toothpaste is white because we put titanium oxide into toothpaste. People aren't even thinking of the basics in terms of what they're controlling. So most of our implant series comes from extraoral. We deliberately moved to things like Baha's and transcutaneous to look at, because we wanted to at least control for known contamination. We do everything metal free. So you know, everything from processing of tissue, you know, taking the tissue out, in terms of how we cut it from a cryotomy, it's all diamond knife. There is no contact with metal. It's a huge amount of effort. Very, very few studies, if any, have reported that level of diligence in trying to do this. And then you come to the picture, and this was a German group who are pretty good, who said, can we look at their data for them? And they've done a similar study on zirconia. And yeah, I mean, it's full of micro debris as well. So I think it's an interesting area, but I go back to what I told the BBC 14 years ago, which is I'm not saying that it's causing implants to fail, and dental implants are still a phenomenal thing made of titanium.
Okay. Thank you.
More questions? Valentin.
So thank you for the very great presentation. I'm looking forward for your study that you show about your students, so to come up with. You mentioned about possible corrosion of implants and so on. If you look back at the literature, you're gonna see that the corrosion can occur by itself because of the pH or whatever, but we also have, for example, some infection that might be caused by the ions or the particles of titanium around the implants, and this can modify the environment. And so, so my question is, do you think if you think about the titanium particles can cause infection, or the infection is causing the release of titanium particles, or is both sides?
So we've looked at this in detail, and so let's go back to the first statement. It should not depassivate. There should be absolutely no spontaneous depassivation of the surface. So the most obvious way is through any connection. So small micromotion at connection, something called mechanically assisted crevice corrosion, which could be between your abutment and the top of the implant. But it can also between, and this has been proven orthopedically, between any slight motion, and we're talking less than 5 microns between mineral and the surface as well, if you have any small detachment. You're now leaning towards cellular-based corrosion, where you have a biofilm, for example, stuck to the surface. Yes, they can trap local chemistry, whether that local chemistry is acidogenic or if that local chemistry is reactive oxygen species. So again, we've done, other people have done, you grow a biofilm on a titanium surface, you take the biofilm off, you do mass spec on the biofilm, and you have more titanium in than you started with. You can do the same with macrophages, you can do the same with osteoclasts, you can do the same with neutrophils. So yeah, there's definitely a cellular mechanism. Whether it explains the distribution or not is more difficult, because it's sort of got to get out into the tissues, and we're talking fair distances. We're talking, you know, millimeters away into the soft tissue space. But yeah, I mean, it's a really, all I say about that area is work with a material scientist and a metallurgist if you're going to report things, because there are fundamentally, the one I hate is titanium ions. Do you know how long a titanium ion will stay as an ion in a physiological condition? We're talking femtoseconds. You know, it's going to oxidize. It cannot. You know, this idea that you have a flux of titanium ions, absolutely not. Titanium is so reactive, so it only can do two things. It can you know, react with water, form an oxide, or it can jump onto a protein or onto some other biomolecule. It's the only two things it can do. There is absolutely, so I mean, there are papers out there where researchers have taken, they say we've stimulated cells in culture with titanium ions. What they've done is taken a titanium ion reference sample for mass spec, so a calibrant, which is in 4 molar HCl. So what they've actually done is poured HCl on their cells, and no wonder something happens to them. So, well, sorry, actually it's nitric rather than HCl. So yeah, they've just done something else and then interpreted it as being this ionic thing. I think it's one of the most dangerous areas in periimplantitis at the moment, because the biologists are not talking to the chemists or the materials chemists on this one. If you can close that and actually write something really sensible together, and I'm fortunate to work with like one of the preeminent corrosion scientists in the UK on this, but even with that, we're so cautious in publishing the story.
Thank you.
I wanted somebody to ask me something about Komodo dragons, because it's much more.
Thank you very much for this very nice presentation. Have you found something about the organic matrix composition and organization of about the study with the enamel of the reptiles?
Yeah, we are working actively on that at the moment, and I just had some images sent to me on my phone. Yeah, so we're doing that from a number of routes. So we're doing it from, because we're in this lucky position to have teeth that we can demineralize and a fresh jaw section. We're trying to work out the cellular mechanisms, but also from a histology perspective. And then from a chemical perspective, yeah, we're starting to do some on the teeth themselves, some vibrational spectroscopy, so mainly Raman at the moment.
No more questions? So thanks again for being here. Such a pleasure to have you here with us. And I hope you can come again.
Who am I passing the baton over to?
It's Valentin. Okay. So the next professor will talk with us is Professor Valentin Barão. You're always welcome, so feel free to stop when I put your presentation on.
Now can you hear me? Yep. So hi everyone. So thank you for being here today. I have to thank Dr. Skel for the very great invitation. So for me, it's a very great pleasure being here at 4P. I did my livre docência here, so I got my associate professor title from 4P. So, and I also have so many professors here that I admire. I'm not gonna say the name of everybody because sometimes I can forget some. But of course, Professor Ricardo, I remember him since I was a student in the masters. He was the fellow of my committee. I don't remember if it was master or PhD, but I think it was master. And Professor Paulo, I always call him because of FAPESP. We have always some problems there, so he helped me quite a lot. So thank you everyone for being here too. So everybody here is from masters and PhD, most of them. No, we have an undergrad student too. So what are you? Oh, postdoc. Okay. So yeah. So today we're going to talk a little bit about biomaterials for dental implants. Mostly we're going to focus on surface treatments for those dental implants, but our main focus here is to develop some antimicrobial strategy to treat the infections that affect those implants. Because as you may know, periimplantitis is a very well-known complication for dental implants. It's the most common biological complication for dental implants. So you need to have some strategy, focus on the biomaterial to overcome such complication. So I think everybody knows that dental implants has been used quite a lot worldwide, not only in Brazil, but worldwide. In US, this number grows about 500,000 per year, the number of dental implants. The implant market is billions and billions worldwide. And why dental implants is so used nowadays? Because it's the easiest way that you can get to health, to natural teeth, because you're gonna eat, smile, laugh, talk, kissing, and enjoy your everyday life. So in other words, according to the American Academy of Implant Dentistry, using dental implants, you're gonna improve the quality of life of several patients, right? But of course, when we talk about dental implants, we need that some surface properties is very important. The most important one that I'm showing here is the biofunctionality. So biofunctionality is the property of the dental implants that are going to make the implant survive against mechanical and chemical actions. And those properties, we can talk a little bit about roughness, wettability, hardness, and resistance. Of course, hardness and resistance is important for the implants do not fracture, but roughness, when the implants were come out on 60s, most of the implants were machined by nature. Remember when Bernini-Marke discovered that titanium cannot integrate it? But nowadays, I would say 100% of the dental implants, they have surface treatments. We can have like treatments that you can add something, or we can just remove something. So subtractive or additive additive methods. But 100% of them that are in the market, they do not focus on antimicrobial effect. They focus on accelerating of bone apposition or something like this, right? And also wettability. You might think that most of the implants, they have a very high hydrophilic surface, but not. If you go to many implant brands worldwide, most of them, they have a very low wettability, which means that they are more hydrophobic than hydrophilic. Of course, you have some brands like SLA Active from Strauma, from Neodent, for example, they have some surfaces that are super hydrophilic, but most of them, they are not. But of course, we are using these implants in the human being, so we need to have biocompatibility. I'm not going to talk about it a little bit. But what is the material that most fulfill the biocompatibility and also the biofunctionality? It's titanium. Until nowadays, titanium is the most common material to manufacture dental implants. Of course, you have some alloys like titanium, aluminum, vanadium, and titanium zirconium that has been used in the market, but most of them, they are commercial available titanium grade 4, according to American Society for Testing Materials. So why titanium has been quite widely used? Because the success rate is still high. If you go to normal patients, like patients with good bone volume, good bone quality, without systemic disease, the success rate is very high. This is high because of the biocompatibility, the non-toxicity, hemocompatibility, osseointegration, and also all the mechanical and chemical properties of these implants. But of course, we have some problems because the oral cavities, we could say that is a complex degradation process that involves mechanical, chemical, and microbiological action. Mechanical, we might have some micro movements in different interface, because although the parts of the implants, they can be screwed or they can be cemented, we might have some micro movements, and these micro movements can promote the degradation of these implants or something like this that would call tribal corrosion or something like this. You might have also fretting corrosion. Chemically, we have saliva fluoride, change in temperature, change in pH. But to make things work, you have microbials. So you have biofilms, you have metabolites, you have acids. And talking about microbials, the implant disease, the dental implant-related disease, or the dental implant-related infection, where you call peri-implant disease, is the most common, as I mentioned at the beginning of my presentation, is the most common biological failure for dental implants. And the main cause of peri-implantitis is the biofilm. Without biofilm, maybe we do not have peri-implantitis. I know that biofilm by itself is not the only one responsible for the peri-implantitis, because you have the host response, the immune response, and so on. But the biofilm will be there, and the biofilm can promote, can trigger this disease. But of course, everybody has biofilm in the oral cavity. If you have dental implants in your oral cavity, you have your teeth, the biofilm is gonna be there. But sometimes this biofilm is under a healthy status. So the disease is not going to happen from today to tomorrow. So the disease is a transition process. In the beginning, you have a healthy status of this biofilm. If you have a dysbiosis of this biofilm, transforming this biofilm from a healthy status to a disease status, we're going to start having the mucositis. Mucositis is the inflammation of the soft tissue. You might have some suppuration, might have some bleeding, but if you can control this biofilm, we're going to reverse to the natural, to the healthy status. So we could say that mucositis is a reversible disease. But if mucositis is not treated, mucositis is going to be transformed in peri-implantitis, because the disease, the infection is going to go from the soft tissue to the bone, and you're going to have bone resorption. And the bone cannot grow by itself. So you're going to need some regenerative process or regeneration process to recover this bone. So we could say that the peri-implantitis is a irreversible condition. So you cannot return to the health status without any treatment and so on. To see how important is the peri-implant disease, we can see that in some meta-analysis studies, the peri-implantitis can account 1 out of 4 implants placed in the patients. And the peri-implant mucositis can account about half of the implants placed in the oral cavity. So the problem is here. And we cannot believe that the problem is very high, but up to now, we do not have a gold standard treatment as you have for periodontitis, for example. Many treatments that you use for periodontitis doesn't work for peri-implantitis. But of course, we have some protocols. We're gonna use like, we're gonna mechanically remove this biofilm. We can use chemical agents or physical agents trying to control this biofilm, but we do not have any gold standard. If you wanna see more information about how to manage this peri-implant disease, I really recommend these articles from Periodontology 2000. Well, in my lab, we have been working with several strategies, most of them focus on dental implants. We have worked with some surface treatments to analyze bioactivity, like calcium-phosphate-based films, bioglass films. We have worked with some basic science, trying to understand how the titanium ions and particles affect the peri-implant area, the peri-implant around the implant. We have been working with titanium and titanium ions, the development of new materials to manufacture those implants. We also have been working with corrosion and mechanical and other mechanical analysis, and mostly we have been working with biofilm-related failures. So we have tried to develop a strategy to control these biofilm-related failures, mainly focus on titanium surface, mainly focus on the surface of the implants. Well, in the oral cavity, we have more than 700 microbial species that can colonize the oral cavity. So, and these microorganisms that is sometimes they are swimming in the saliva, they can aggregate between each other in the saliva, but can also adhere on surface. They can adhere in biotic and abiotic surface. So if you have a removable partial denture in the oral cavity, you're going to have biofilm in the acrylic resin, you're going to have a biofilm in the cobalt chromium alloy infrastructure, we're going to have the biofilm in the tongue, in the mucosa, and so on. So the biofilm is going to be there. And of course, the biofilm to be formed, we have a first biological response. And this first biological response is the adsorption of proteins. So as a simple way, we have like here the titanium, that is the titanium implant. We have the proteins from saliva or the proteins from the bloody, and this protein is going to be absorbed on the surface. And this absorbed layer have some links to adhere microbials that are swimming in the oral cavity. So understanding the proteomic profile of this surface is very important to try to develop a new strategy to control this biofilm formation. So in this study that is still under review, we hope that it gets accepted soon, we try to investigate how the absorbed proteins is different when you compare titanium, animal, and dentine. And in this Vance map, you can clearly see that if you have the source of proteins, the saliva, we have 42 proteins that was only absorbed in dentine, 3 that were only absorbed in animal, and 12 that were only absorbed on titanium. In the plasma, we also observe something similar. So in other words, I don't know if this might explain why in some cases the one treatment can work for periodontitis, but doesn't work for periimplantitis, because the substrate of the biofilm that adhere on the surface is different. The protein layer is also different. And we also run some microbiome analysis, and this I know that is scary a little bit, it's scary for me too, but here we are showing that if you compare, for example, titanium and dentine, in terms of the composition of the biofilm, if you go to red, is the pathway of the bacteria, the species of bacteria that are more evidenced in titanium. If it goes in blue, is the species that is going more for dentine. So the same source of the biofilm, only changing the substrate, the composition is also very different. So if you compare titanium dentine and titanium and animal. We also checked for the same substrate. We're using only titanium, but compare commercially pure titanium, titanium aluminum vanadium, and also some experimental titanium alloys, titanium niobium zirconia. We also observed some difference in terms of protein absorption. Using the same material, titanium zirconia, for example, only changing the surface profile, only changing the surface composition. Here we use machined, and here we use SLA surface from Straumann. As you can see, for saliva and for plasma protein source, we have some specific proteins adhered only in the machine, and the others only in the SLA surface. And these also modulated the number of bacteria adhered onto the surface. So the formation of this biofilm and the disease progression is very complex. Here, this scheme shows how the biofilm can be formed on the titanium surface. Of course, the event's not going to occur like isolated, as I'm showing here. Everything is happening in the same time. But to understand, in the beginning, we're going to have the protein layer adsorption. Then the bacteria that are planktonic. We know that some bacteria, they cannot be only planktonic in the saliva, because Professor Kuhl, he's a Brazilian that is in the University of Pennsylvania, he's already showed that we have some biofilms in the saliva. So some bacteria are there between each other in the saliva. So we might have some microcolonies on the saliva. So these microcolonies or the bacteria isolated can adhere onto this protein layer, starting from forming these microcolonies, and we're gonna have a process of co-aggregation. But the bacteria, the biofilm is a 3D structure composed of bacteria, virus, and so on, whatever, fungus, and so on. But it's also composed of a extracellular matrix. And this extracellular matrix works as a shield, a protection for this biofilm. It's going to give the structure, the 3D structure, but it's going to also protect the biofilm. Because when you use drugs, for example, antibiotics, if you have a very robust biofilm matrix, the antibiotics has some difficulty to penetrate in this biofilm. So this biofilm matrix has been also a target to control the biofilm formation. Some drugs can be used to degrade this biofilm matrix. If you degrade the biofilm matrix, the biofilm is going to be more susceptible to any other antimicrobial. But yes, we have this very complex biofilm format. In the beginning, this biofilm is more acidic, we call that. So the pH in the biofilm tends to be low, but as the time goes by, the level of oxygen tends to be reduced because the biofilm is very thick. So in the bottom part, especially in the bottom part, we're gonna have the survivability of those bacteria that doesn't like oxygen, like the red complex bacteria that are related to the infections, the peri-implant infection or the periodontal infections. And also during this whole process, we have some bacteria that are detaching from this biofilm and forming again this whole process. Of course, when we talk about implant surface that degrade, that promotes antimicrobial effect, we are not saying that we're gonna destroy the biofilm, because it's impossible to have a surface-free biofilm. So we are thinking about having a surface that can control this formation, this biosis condition that comes from the health to the pathogenic one, right? If you talk a little bit about the mechanism on how the surface of dental implants can control this biofilm formation. As you may see here, we have a microstructure surface, a macro, micro, and nano structure. Is very tricky for Portuguese and English for us, because this is micro, it's in Portuguese and English is very difficult. So this is the size of the bacteria, 1 micrometer, for example. If you have a macro structure, we're gonna have a very long way so that bacteria can adhere on the surface. If you have a micro, we're gonna reduce. We might increase the total surface area, but the active surface area is being reduced, especially in the nanostructure. As you can see here, we have a very long area, very high area, but the active area for the bacteria to adhere is very low. So we have in the literature several nanostructure surfaces that can repel the bacteria by itself without having any antimicrobial. In terms of coatings, we have different techniques that we call bottom-up or top-down, or the combination of both. The bottom-up is the formation of this coating from the bottom. For example, anodization, plasma electrolytic oxidation. We're gonna oxide the surface. We're gonna oxide the surface from the bottom, and then a thick oxide layer gonna be formed. This is very interesting because if you are not depositing something, the adhesion between the coating and the substrate is very high. Tends to be higher than when we deposit it, because when we insert the implant to the bone, you're going to have the shear force. If you have some coating that can delaminate, that might not be so good. Or we can also incorporate some antimicrobials and so on. And here's are the main mechanisms. The simplest mechanism is the antifouling mechanism. What is the antifouling mechanism? The bacteria are gonna see the surface, the bacteria are gonna try to adhere on the surface, and then the bacteria is repelled. It's like the Teflon pans. Like if you are frying an egg and the egg doesn't stick onto the pan, right? So you have a Teflon. Teflon is hydrophobic. Teflon is repelling. It's the same mechanism. It's similar. We also have contact killing mechanism. For example, we have some surface that is nanostructure. The surface is very pointed. When the bacteria adhere onto the surface, the membrane of the bacteria can be perforated. So it's a contact killing mechanism. But the contact killing mechanism by itself is not so interesting, because we already see in the literature that if you have a dead bacteria onto the surface, the bacteria is there, it has not been removed. We might have more live bacteria adhering in these dead bacteria. Also have some releasing mechanisms. These releasing mechanisms has also some drawbacks, because the perimplantitis is a chronic disease. If you have a release mechanism that is not controlled, we might have this release of drug in the beginning when the implant is placed, but the disease is not there. I know you can control the initial infection during the healing process, but we might not control the disease when you need. So why nowadays in the literature you're gonna see some the smart or the intelligent. I know some people, they do not like this title, smart or intelligent, but is smart. Why is it smart? Because we can control when the drug is gonna be released. I also make some comparison. In the past, you guys are here very young, but you have other people here that are older than me, but I'm in the middle age, I would say. In the past, when I was like an adolescent, like 13 years, 14 years, if I want to see a movie, I needed to go to a place to rent this movie, right? It's a taper. How can I say video cassette in English? I don't know. You know, you got it? So you need to go there. But you see, for example, it's Sunday night. I want to see a movie. Can I see a movie by that time? If it's not appearing in the TV? No, because it's not on my demand. It's in demand of the store that rent the taper. So this is the normal drug delivery systems of the surface. But nowadays, if the intelligent surface, we call on-demand surface. When you need, we go. So today, or if it's 12, it's 1 in the morning, I want to see a movie, I can go to any streaming, right? So Netflix and so on. So it's all your own. It's in your own demand. So you decide whatever you want to see. So the smart surface might be like Netflix surface, right? Did you get the comparison? It's like this. So I'm showing now. I forgot to say that I have here the photo of the main student that run these experiments or run the paper, and also the QR code, so you can just use your cell phone, and then you're gonna get the paper in full. So this paper, we develop a one-step super hydrophobic coating. Of course, if you go to the literature, you're gonna see several hydrophobic surface, but by that time, we didn't found any surface that was super hydrophobic by just one step. So in this case, we use a plasma surface treatment in an environment containing oxygen and argon and hexamethyldisilloxane, and we develop a very rough surface only with one step. We do not need to rough the surface before. Only the plasma surface by itself promote this cauliflower surface. So as you can see here is the control, no surface treatment, and the cauliflower surface treatment. As you also see, the contact angle between the surface and the water is higher than 150 degrees, which means that the surface is super hydrophobic. In this paper, we're gonna see several cellular experiments, but my focus here is the biofilm. As you can see here, we use a model that tries to resemble the composition of a complex biofilm. We are not using one specific bacteria species, we are using a polymicrobial biofilm. So here's not biofilm because it's zone adhesion. As you can see, the control has more adhesion of this polymicrobial species. We also run for some candida. Why we are using a fungus for perimplantitis? I know that candida by itself, some people, they believe that that is a problem for perimplantitis, but others, they don't. But it might have one clinical implication because the interaction between candida and streptococcus may promote more invasion. So it's a problem. So it's going to have more invasion of the tissue. So why we also checked for candida, and even for a fungus, we observe lower adhesion of this fungus. We also checked for streptococcus mutans. Streptococcus mutans, there's no relation with perimplantitis, but the streptococcus mutans by itself is a very sticky biofilm because they have a very strong extracellular polysaccharide matrix. So using this stick biofilm, we also observed a lower content of Streptococcus mutans in the superhydrophobic. We also check the biofilm 24 hours. We observe lower levels in the superhydrophobic. We also checked in confocal images. You can clearly see in green are the biofilm, the polymicro biofilm. In the experimental, we have a lower content, and this is the quantitative data of this confocal. We also checked if you treat this biofilm here in control, and here in the superhydrophobic, with chlorhexidina, that's a very common antimicrobial agent used to control periimplantitis, we can see that the effect was more prevalent in the superhydrophobic surface. We also checked some drug delivery surface, but this is not under control. We just develop a surface coating containing copper, different sources of copper, as copper acetate, copper sulfate, and copper oxide, using plasma electrolytic oxidation, as you can see here. And also they use the control as the calcium phosphorus, only the control. All the surface we observe the volcano-like surface, like these pores in different size and different numbers, depend on the electrical conductivity of the solution that we use to prepare this coating. The number of pores and the size of these pores varies quite a lot across the groups. We observe that the surface roughness of all these groups is in a good range from 0.7 to 1.2 micrometer. The surface areas tends to increase with the increase in surface roughness, and now surfaces were hydrophilic, as you can see, from 10 to 12 to 18 degrees with the water. But the interesting thing is here, the antimicrobial effect. At 24 hours of biofilm formation, we can see, I would say that's a good antimicrobial effect is like 2 logs of reduction. But when you go to a more mature biofilm, 24 hours, the antimicrobial effect was reduced quite a lot. So this is the problem of those surfaces. We might have a good effect at the beginning, but only after 2 days, there's no more effect. Imagine if I keep this surface in a long term, no effect in this long term. So, but here's the results. Yeah, we have here, but this is not, I think this is not the focus that you should have when you are talking about antimicrobial surface. We also have other surface. In this surface, we try to mimic the bioglass in the surface. We compare with the SLA surface, the commercial available one, in terms of microbial adhesion, microbial formation. But the most interesting results here is the modulation of this biofilm. If you compare in the microbial adhesion, so it's very hard to choose this, we're gonna see that the number or the level of red complex bacteria in the PUBG, we call a plasma electrolytic oxidation containing the bioglass, was lower when you compare to the control. So you might have some surface that can modulate the composition of this biofilm. But my focus here is gonna be about photocatalysis. So as I mentioned, the on-demand surface might be the focus of the future antimicrobial surface. So photocatalysis, so Ana Paula is here, I'm not gonna, she might know quite a lot about this, but the photocatalysis is like, here's the titanium dioxide, for example, is a molecule of the titanium dioxide. Photocatalysis only works when you have titanium dioxide, for example. In the anatase form, you have anatase, rutile, brochite, and so on, but it's gonna work better in anatase. So if you see the structure of this molecule, we have the valence band and the conduction band. When we exposure this molecule to a UV light, for example, we're gonna have the flow of electrons from this valence band to the conduction band, and then we're gonna have the formation at the end of reactive oxygen species. So we have a distance between this valence band and this conduction band, and this distance might be 3.2 electron volts. So it works with UV light. If you wanna have an effect with visible light, for example, we need to reduce this band gap. And we're gonna talk a little bit about that. But of course, photocatalysis has been quite well used for air purification, for water treatment, for self-cleaning surface, for sterilization. And in 2019, we published a paper trying to use this photocatalysis to control the biofilm in dental implant. And well, but we cannot use UV light to treat in the oral cavity. Of course, if you have some, like an overdenture, for example, you have a prosthesis, or you have the metal that you can remove from the patient's oral cavity, you can apply the UV light outside. But if you want to use in dental implants, you cannot remove the dental implants and treat outside and put again, so it's impossible. So you need something that a light source that can be applied directly in the patient's oral cavity. So how can you have this photocatalytic effect under visible light? As I mentioned, we need to reduce this band gap here, that is normally 3.2 electron volts, so it is stimulated by the UV light. But to have a stimulation under visible light, you need to have a distance lower than 3.0 electron volts. You might have in the literature several strategies to provide this effect under visible light using transition methods or using semiconductors. So this is the first study that we published using this photocatalytic effect under visible light. I didn't show the main results of this paper because in 2019, we used this technique to have an antimicrobial effect, but the effect was not so high. But it was our first study, so the effect was like 1 log reduction. In terms of antimicrobial effects, not so high. We also have to apply the light for 30 minutes, so that is too long. Well, but we move forward, we move, and now we have some data that is not published yet. It's from my same student from, this is some results of her PhD study. I'm not showing all the results, but some. As you can see here, we call in the literature as a pancake structure. I cannot see pancake here. Can you see pancake? No, but the thing here is like, because we are packing like pancake, but for me it's not pancake. But the description, the literature is like pancake when you have CO2 by itself and you have bismuth. We incorporate bismuth to reduce this band gap. The roughness of the treated surface, we use a plasma electrolytic oxidation technique. So the roughness is around 3 for the plasma electrolytic oxidation surface. We were able to deposit anatase and rutile structure in the TO2 and bismuth TO2. The water contact angle was very low for the treated surface here, super hydrophilic. We also have the experiment showing the release of the bismuth. Of course, we have a burst release in the beginning, but this release was sustained a little bit. And then you have also some data about hardness, some data about friction coefficient, some data about tribal corrosion. As you can see here, this is the drop of the potential when we are rubbing. As you may see, in the controls, SLA and machined surface, the drop was higher when you compare the TO2 surface, which means that in terms of tribal corrosion, the TO2 surface were better. We also have some white light interferon microscope image. We also have the material loss after rubbing. The material loss was lower for the bismuth and for TO2. We also have the photocatalytic effect under visible light. As you can see in different techniques, we observe a greater photocatalytic action for the titanium bismuth. When you have the bismuth, the effect was higher. We also, I'm sorry because the image is not so high, but we use different techniques. We use different biofilm models to verify the antimicrobial effect. In that first study, we use a model with only 2 bacteria that does not resemble what happens in peri-implantitis. But here, we also run, again, using the same bacteria in the beginning, but we go further. We use a microcosm model that we use saliva as an inoculum, and we have a grow condition using an aerobic and anaerobic condition. I'm going to show in the end of my presentation this model. And then at the end, you have a biofilm composition that is similar to the peri-implantitis. And we have an effect under visible light, especially for that group that has a photocatalytic action under this visible light. We also run some experiment in situ. We use a palatal appliance in patients, and this patient's gonna use this palatal appliance from 3 to 7 days. I don't remember in this study if it was 3 or 7. We use normally 3 or 7. And then we apply the light at the end, and even in the in situ model that tried to resemble this composition, we also observed a good effect. We also tested a recolonization of this surface when we do not, after light exposure, the recolonization was not higher. So you might have no alteration on the surface that might promote greater bacterial attachment again. We also run some in vitro experiments using cells. Let me see if I can. Oh, it's not working. How can I go here? Let me see. So as you can see here, the CCK8 using perosteoblastic cell doesn't have any effect, any negative effect. We also have this nanolive use exposure in these cells to bismuth, and it seems that they are pretty similar when you compare the control without bismuth and you have the bismuth into the cell culture. We also investigated some gene expressions, RUNX2 and collagen type 1. We observe a greater content for the RUNX2 and this collagen for bismuth. We also run some in vivo experiment using in vivo infection. So for this, we use a microbial contamination using the in vitro model that I'm going to show later. And these discs were submitted to the dorsal of the rats. And then after light exposure and during 3 days, the rats were sacrificed, and we count the number of bacterial cells on this infected tissue. As you can see, the surface that were light exposure, we observed a reduction in terms of contamination, especially for the bismuth that has a photocatalytic effect under visible light. We also observed a reduction in terms of inflammatory cells, as you can see here in this graph, counted in this graph. We also checked the bone formation using this in vivo, using in vivo analysis. As you can see, we use the tibia of these rats with the surface treated with bismuth and SLA. SLA we use it because SLA is a very common surface treatment for dental implants. As you might see, in terms of micro CT, there's no much difference across the groups, which might be very good because we are not looking for something that is better than SLA. We are looking for something that is very good in terms of antimicrobial effect, because normally if you have a surface that has a great potential, great antimicrobial potential, normally the surface might be cytotoxic. So if you have a surface that at least is the same as the one clinically used, it's very good for us. We also checked for some histology images in terms of new bone format, in terms of collagen. Some data were much better for the bismuth CO2, and here's some gene expression that we also observed better expression, upregulation for those in the bismuth surface. We also have developed other strategy, like in this study from my PhD, my previous PhD student. So we developed here a molecular imprint polymer that you can deliver folate. So folate has been used in the past quite a lot for periodontitis, because it can control the inflammation. So people normally administer that systemically, but in this case here, we try to develop something that can be released locally and controlled by the pH. So we were able, I'm not showing the data here, but just the image here, we were able to show for the first time that fibroblasts, they have a receptor. So inflamed fibroblasts, they have a receptor, the FOLR1 and FOLR2 for this folate, and this folate can be penetrate into the cell and promote the anti-inflammatory effect. So if you go to this article that's already published, you're gonna see several data we have since from the development to the in vitro and in vivo analysis, showing the mechanism, the antimicrobial, not the antimicrobial, but the anti-inflammatory mechanism. In the beginning, this is a FAPESP supported study, but in the beginning, our main aim was to develop a MIP, a molecular imprint polymer, that might have the anti-inflammatory effect, but also might have a degradation effect in the biofilm matrix. But we tried several strategies, but we were not able to develop this MIP. We have a collaboration with the chemistry from Araraquara, Professor Maria Sotomayor. I don't know if you know her. Yeah, she's from chemistry. So yeah, so this is interesting because Our aim here is not to have a surface treatment. It's like to have a biomaterial that can apply directly into the pet implant area. But of course, our data here is just in vitro and in vivo using animal model. We do not have any clinical data, but we don't know if you put this clinically, it can work as a foreign body. For example, I don't know if you're gonna may induce more inflammation, more response, immune response. We don't know, so we needed to go further. But until now, our data was very interesting. We also developed some hydrogels that you can. This data is not published yet. It's also from my previous PhD student. We developed with the people from chemistry too in UNESP, Araraquara. We developed a thermal-responsive hydrogel. So this hydrogel, when you have 34, 35, or a little bit higher, we have the gel formation, as you can see here. We use PNVCl, and you incorporate tetracycline, and we were able to have a drug delivery system using this tetracycline. We use an infection model we also have some in vitro analysis, but you have the in vivo that we show that the bone formation in the group that was treated with tetracycline and this hydrogel was better. We also have some data in terms of microbial composition. So we also observe some better in terms of this for a healthy status when you compare to the non-content of tetracycline. So just to show you here, I think, yeah, I'm the time. We have been using quite a lot this model, this in vitro model, to have a biofilm that can resemble the composition of the perimplantitis. We also run some experiments comparing this biofilm with the clinical data from studies that has that collect the biofilm from perimplantitis area, and the composition is pretty similar. So in this model, we run the single model up to 144 hours. It's not just 2, 3, 24 days, it's several days. So we have different culture medium, we have different culture conditions using, of course, in this phase is aerobic and this phase is anaerobic. Here we are trying to replicate what happens clinically, because in the beginning, the biofilm is gonna adhere onto the supragingival area, then the subgingival area gonna occur afterwards. So here is the process of the transition from a health status to a pathogenic status. We also check it because every single time I send grant to FAPESP, I'm always asked about this, and I'm show the data, I'm show the papers, but reviewers still keep asking every single time. I don't know, the microbiologist or the periodontist or the implantologist, I don't know. But we also, from my students, is published already, we compare, because normally I use saliva from health patients. I do not use saliva from patients with periimplantitis. And it's pretty similar. Why we use from health patients? Because the students needed to run the experiment in the morning, or like 6 in the morning, 7 in the morning, and they collect the saliva by itself. They collect their saliva. They collect the saliva of their colleagues. So if I need to collect saliva from patients with periimplantitis, I know it's hard to get these patients. We're gonna need to store the saliva, and normally we want to standardize the time that the saliva is collected. So the time that you're going to use, we want to choose fresh saliva. So it's much harder. So in this study, we have like saliva as inoculum from the saliva of patients, normal patients, no disease patient. We also have saliva, and then during the biofilm formation, we add some red complex bacteria to see if you're going to have more expression of this bacteria. And also we collect biofilm inoculum from the patients with periimplantitis. And surprisingly or not, using that model, we have pretty similar composition of all these three biofilm. And we also have, in this study, we get some data from my colleague, Jamil Shibli. He works with some, he has several clinical data. We got those data from him, and then we met the composition of this clinical data with these biofilms. And using saliva inoculum is enough. So even though I show that, people are gonna request me other analysis, I don't know. So we also use quite a lot in situ models. We have some To use this in situ model, we use sucrose 4 times a day. Why we use sucrose? Because sucrose is going to stimulate the formation of biofilm matrix, and you have the more biofilm matrix in there, the more we're going to have the transition between the health to the pathogenic bacteria. So we already have some studies showing this protocol in Journal Periodontology. We are also using this animal model because we do not have a very standardized animal model to have the perimplantitis. Because I know the perimplantitis is in an open condition, is in the oral cavity. We have some model, not me, but in the literature, you're gonna see some models that use the oral cavity of the rats as a perimplantitis model, but it's very, very, very difficult to run because most of the implants, they are loose. I know a Brazilian professor from UCLA, Flávia Piri, I don't know if you heard about. She's famous using this model, but even her, she has so many problems running these models. And many models in the literature, they use rats, tibia, for example, and they use, for the infection, they use inoculum. They get the bacteria inoculum, and then they place there the inoculum. But the inoculum by itself is not a biofilm. It's only planktonic cells. The inoculum by itself is not a complex biofilm because it's only one species. So in this study, we culture the first threads of the implants in vitro using that model, in vitro model, an aerobic and anaerobic. So we have here a very complex biofilm with several species that resemble the perimplantitis. And then we create a defect in the rats, and then we place this infected implant in the rat. Of course, we have to remove mechanically part of this biofilm, otherwise the rat's gonna die, because you do not want that, or the implant's gonna be loosed, because you do not want that the implant loosed by itself. You need to have the infection there to test our antimicrobial agent or so on. So this model we use for that one in the hydrogel model. So yeah, even though you have several strategy, if you go back to the literature, you're gonna see so many strategy. We do not have filling this gap. Well, we have very scientific development in terms of antimicrobial effects, especially for surface treatments for dental implants, but we do not have the commercial development of this surface. So many drawbacks might be answered, like the controlled release, the difficulty in control the cytotoxic effect, the difficulty in developing those surfaces, the long-term stability of the surface, and so on. So it's in my dream to have something that's in the market in the future, to have, I think the people that discover this or put this in the market in the future are gonna be very rich. So hopefully it can be me or some person nearby me. Here I show you some, mostly here, I think 100% here are only literature review papers from our group. So if you wanna see a little bit more about infection in dental implants, how to treat the surface and so on. So, and also this one, the last one was about hydrogels. So you can go in these publications. Of course, all these studies is an interdisciplinary study. I'm a dentist, so I do not have all the facilities, I do not have all the knowledge. So the combination between the dentist or the chemistry, the cell biologists, people with microbiology understanding, it's very important to create a very robust research project. So as a take-home message, I would like to say that the dental implants is still a viable alternative to replace missing teeth. We might have the perimplantitis being affecting the longevity of these dental implants. We still have some space available to develop smart coatings or other strategy to control this disease. And it is still necessary to bridge the gap between the science and the industry. So I would like to thank all my students, not all are here, but nowadays I have 90 PhD students, 4 master students, and 3 postdocs, so we are very big now. but without them, I cannot run those experiments. We are starting our Instagram group that is called BAN Group, that is Biomaterials and Associated Infection Network. If you follow us, I thank you. And we use it to share our publications and so on. Everything is in English because we wanna to cross the ocean. We wanna go outside the country. And then I would like to give you one message. I know that my message is you need to challenge yourself and step out of your comfort zone. And I put here some words about Chinese, 我学习中文, because I'm learning Mandarin. I'm trying to learn Mandarin, so I challenge myself every day because it's very, very difficult. But I've been in China with a collaboration project with CNPq 1 year something ago, a year and a half, and I feel in love with China. So I decided when I came back, I would start learning Chinese. So I started this year. I'm paying another course, but I'm not doing, but now I'm doing other. So, but every day I study a little bit about Chinese, so I challenge myself. So everybody should challenge yourself in some part of your life. Can be in research or can be in other parts of your life. So I'm trying to challenge, in all my studies, I challenge myself. So I'm challenging myself in other parts of my life too. So this is the message that I would like to share with you today. And also, of course, I have to, oh, it's not appear here, it's 谢谢. 谢谢 is obrigado in Chinese. I have to thank, of course, all the Brazilian supporters, FAPESP especially, and also CNPq, and CAPES for providing also some grants for the students, all my collaborators from Brazil and outside the country. So thank you very much. I'm ready to entertain any question that you might have. Thank you.
Thank you, Valentin, for your beautiful presentation.
Thank you.
So we are open for the questions. Who want to ask?
Valentin, congratulations on your presentation. Thank you for your presentation. And you talked about hydrophilicity, right? And you use water. And when you use blood and serum, it could be different, right? I mean,
yeah, might be different. Of course, if you go to surface-free energy, we have your other polar and apolar liquids to have the as a standard.
Also, yeah.
But we have been using water because it's also very standardized in the literature. But of course, it's different.
Yeah,
I know that one.
I think material scientists prefer to use water.
Yeah, because if you need blood, you need to collect blood. You're gonna need to have the IRB, or you can use the blood from those animals that you use for. I forgot the name in English. Forgot. Even in Portuguese, I forgot the name.
For all your studies, you use water. I
mean, for water contact angle.
Because sometimes I have some differences in terms of wettability.
Yeah.
Because we cannot use hydrophilicity for blood, right? So. Yeah. But it's okay. Water.
Yeah, water. Yeah, okay. Water.
Thank you.
Valentin, fantastic work.
Thank you.
Many congratulations. I want to touch on what you said right at the end, which is this first translational gap that we all face, trying to convince somebody to take forwards an innovation into practice. And I know particularly in medical devices, there's this question of surfaces that are designed to be preventative versus surfaces that are designed to be therapeutic.
Yes.
Do you think we will ever get to surfaces that are designed to be therapeutic into practice?
No, it's very difficult.
Yeah, I think as much as the smart materials and so forth are,
because it must be much longer term.
Yeah. So, out of the work that you're doing, which direction do you think is most likely to lead you to
preventive? Preventive. Yeah, well, if I talk about photocatalysis, can be preventive because imagine we have some tests that we did a cycle. We run the experiment several times. We run today and then we run another day to see if the photocatalysis is maintained in the long term. So think about the clinical application when the patient comes to the office, and then we can apply again. So to prevent the infection or something like this. So as a preventive measure.
I hope you can do that.
I really hope so.
I really do.
I have to say that it was funny when you said about photocatalysis, because Ana Paula is here, and so. But
I'm afraid of you.
No, think about my situation now. I have to talk about teeth and bones to hundreds of dentists.
Yeah.
It's much more difficult. And I was thinking about the surface that you modify. You showed mainly a result about contact angle, just to say if the surface is hydrophilic or hydrophobic. Have you measured the surface energy?
Yeah, but not in this study. But in my previous studies, we always measure the surface energy using polar and apolar liquids.
Because, for example, you said about Teflon. Teflon actually is not hydrophobic, nor hydrophilic.
Okay.
Teflon.
So I was wrong every time.
No. No, I will correct you what I said. So it's hydrophobic and hydrophilic, both.
Depend on the liquid.
No, because it has a very low surface energy. Because when we wash the Teflon dish, we see that the oil doesn't attach at the surface, but even the water.
I got it.
And usually we think, oh, what is hydrophobic is not hydrophilic, what's hydrophilic is not. In the case of Teflon, no, it's not hydrophobic and not hydrophilic, because it is called inert because of the surface energy. The surface energy is very low. So water, polar and non-polar compounds doesn't attach on this kind of surface. Why I'm saying this? Because the result showed about the difference in the attachment on the surface of dentin, enamel, and titanium. Obviously, we have a different surface chemistry, especially when we are talking about titanium and the other tissue and the tissues. But we have also a difference in the surface energy.
Yeah, the reviewer, you are the reviewer of this paper? No. The reviewer asked us to run this experiment too. So you are running for titanium.
And it's very interesting because you can have surface with the same surface energy, but if you have different components, polar and non-polar, the attachment of proteins will be completely different, even if you have the same surface energy. So it's very interesting, subject to go in depth. And it was very nice. I think I will be a candidate to go to work in your group.
Now we are working together, João. Yeah. Thank you.
Thank you, everybody, for the questions. And thank you again, Valentin, for coming here. I'm so glad to have you here, too.
Thank you.
Now, the last presentation is from Professor Ana Paula. You're free to talk.
So, thank you very much for the invitation to be here today. I love coming to the dentist school. I was talking to my colleagues that I feel like home when I'm here. And I really like to collaborate with you, to talk, to share, because I know that the things I do, like chemistry, I can only see an application when I collaborate with people that is working in clinics. And so it's very challenging to me to talk about teeth and bone with this audience. And so today I will, to finish my presentation. So that's the end, and we can eat now. Deixa eu ver o que eu fiz aqui. Ok, onde que é a ponta? A ponta é aqui. Você tem que ficar segurando. Ah, tá bom. Deixa eu ver. Okay. So, I am the coordinator of the Laboratory of Physical Chemistry of Colloids and Surface at the Faculty of Philosophy, Science and Letters at Ribeirão Preto, here in this campus. And today I will talk about how the nature can inspire us to prepare biofunctional materials. So I will start with the definition of biomimetics. Biomimetics is the field of science in which inspiration are elicited from nature to design practical materials and systems that can imitate structure and function of native biological systems. There are some important points in this definition. First, biomimetics is not only to imitate, to replicate morphologies. We, in the biomimetic field, we replicate morphologies to get functional and advanced materials based in the native system. So if I have a cup, but if it doesn't work like the cup, it's not a real biomimetic system. So based on that, I can give you some examples about the nature and animals that can be inspiration to produce functional surface like superhydrophobic functional surface, like Professor Valentin was talking about, with this kind of nano patterns that can repel water. And this surface can be inspired in the feet of this kind of mosquitoes that doesn't penetrate water. We can also get inspiration from these butterflies, from the flies of the butterflies, to produce optically functional surface. The smart adhesive surface inspired in the gecko feet, that's classical. You know also that clothes that the swimmers use are also based in the skin of sharks. We can also produce functional surface to capture water based in the structure of feet of some animals. And we have some intersection among the definitions of biomimetics, biomorphism, and bioutilization. So we see that we have to replicate and to try to use. So what's the inspiration that I got and my group get from nature? Bone. Bone is an inspiration for us to produce materials with superior properties to stimulate bone repair and bone formation. So before talking about the materials we prepare, and if I want to imitate something, if I want to replicate a structure, I really need to know what is this structure. And to get bone as an inspiration, I know that I can try to mimic first the organic part of the bone using lipids and proteins. I can try to mimic the mineral part of the bones, trying to produce some substituted apatite. I will talk today a little bit about a special kind of substitution in apatite where calcium can be replaced by strontium. I will talk about the role of glycosaminoglycans in the replication of the bone structure. And I'm not talking more about the starch-based membrane because I don't have time, but I can share another opportunity with you. We have to also keep in mind that I can mimic the structure of bone, but bone is not useful in our body structure only for mechanical protection. We have to keep in mind that bones are the main ionic reservoir that we have in our body. And it's responsible for controlling the release of ions that are important for the most part of our biological functions. Trying very, very quickly to understand the bone structure, we can understand bone as a nanomaterial if we go from a macro and a micro to a nano scale. So from a micro scale, the bond is organized to a micro scale in compact bond and trabecular bond. The structure of apatite and collagen is ordered in a lamellar way, in a hierarchical structure, based in a cholesteric organization of collagen fibers. The organization of these fibers gives origin to these, here I'm showing in dark blue, these D spaces where a mineral can penetrate and mineralize intra and extra in inter and extrafibrillar way. Looking at the collagen structure, it is organized in a triple helix composed by the proteins and then the amino acids. If you split the structure of the mineralized collagen fibril, so we can find this hierarchical, very ordered structure of collagen and apatite in this kind of needle-like nanoparticles. So first, I will talk to you how we can mimic the organic matrix of the bond using a model called Langmuir monolayers that is useful to replicate the cell membrane, the structure of the cell membrane bilayer. Using lipids and equipment called Langmuir Truth, I can organize lipids at the liquid-air interface, trying to resemble the structure of the cell bilayer, and I can also attach proteins and enzymes like alkaline phosphatase to try to biomimetically generate phosphate from the hydrolysis of ATP. I can also transfer these monolayers containing or not enzymes and proteins to titanium surface to produce bioactive coatings on these implants. But why I want to create a modified surface of implants with this kind of biomimetic films? If I add a structure similar to the structure we have in the cell membrane, the cells will recognize here not a titanium oxide surface at the first contact, but the cells will recognize a more chemically and physically similar structure after the first contact with the surface of after the contact with the surface of the implant. So it's a more biologically friendly surface for the cells to attach. And it was our first hypothesis when we started working with this kind of modification, that cells, after recognizing a biologically friendly environment, could could replicate better and mineralize better when we're talking about osteoblasts and odontoblasts. Then, creating this interface, we hypothesize that the attachment of the implants into the bone tissue would be enhanced. We can play with lipids with different structure, like lipids only one carbon tail, lipids with a double carbon tails. We can play with a surface with higher affinity by calcium, with lower affinity by calcium. We can play with the number of layers that we are depositing on titanium. And as a first example of this kind of films and coatings, I will show up the result of a paper that we recently published in collaboration with Skell and Professor Owen. In this study, we coated the surface of these mini implants using these lipid films, and we transferred calcium and strontium to the surface of the lipids Then we mineralized the samples after the contact with a calcium phosphate, a phosphate buffer, and a simulated body fluid. We implanted this mini implants in the bones of osteoporotic mice model with collaboration with colleagues from UNESP Aracatuba. And we have lots of results, but I selected this result as an important one. The coated surfaces had a higher force, reverse torque, needed to be removed from the bones of osteoporotic mice compared to the control, that is the uncoated titanium. And if we see here, the effect of strontium here, we had 90% of strontium at the surface of the implants, 10% compared to the amount of calcium. With strontium, the implants were better attached at the surface. And the effect of strontium was more important in osteoporotic than in healthy mice. But if you see, even in the healthy mice, the performance of the implants under the reverse torque assay was better. Also, the nail bone formation that we show here by this confocal image, the green area corresponds to the nail bone formation area. Here up, we show the surface of the titanium implants implanted in the health animals, and below, not working well, below, in the osteoporotic model, we see that in increasing the amount of strontium, we increased the nail bone formation. Obviously, in the osteoporotic mice, the bone formation was slower, but we see here that the presence of strontium was important to induce nail bone formation. We can improve this model also adding proteins to the surface of the phospholipid films. In this study, we modified the surface with the lipid films, and we added also collagen to the structure. We compared the composition of the coatings with the composition of a natural bone, a fresh bone, and we see that this kind of coatings composed by the needle-like particles were very similar by FTIR analysis to the bands we find in the natural bone. Moreover, the films containing collagen were much more similar to the natural bone than the films obtained in the absence of collagen. We studied the performance of osteoblasts on this surface, and this is one example that I said this morning. When we got the cell viability results, the cells were much more viable using this lipid, this OPA lipids, than using this GHP lipid. The difference in the structure of these lipids is that OPA has one carbon chain and GHP has two polar chains. So the surface of OPA was more hydrophilic. So the cell viability was higher, and we were expecting that the osteoblasts would work better in the surface. I'd like to go back, wait. But what we found was not, even with reduced cell viability, but higher than 80%, the cells mineralized better in the GHP surface. So what I said to the students in the morning, okay, I got a surface with a very high cell viability. It doesn't mean that the surface will mineralize better. If you're studying mineralization, we should go further with the experiments. MTT assay is not enough. I mean, it is just enough to say if it's cytotoxic or not. You can say nothing more about the surface. We use it like here. It's just another example. We use it also this kind of films not only to deposit calcium phosphate on the surface of titanium, but we also can use this to deposit carbonates. We can measure the surface energy of the surface after the deposition of the films. We can also track the release of ions by the surface. We can use this kind of films also to deposit, after depositing collagen, we can deposit these very beautiful nanotubes composed by calcium carbonate. So more than a model, these bilayers are very versatile to produce biomaterials, functional biomaterials. Okay, I showed you somehow how we can replicate the organic part of the bonds. Now I would like to show you how we can replicate the inorganic part of the bond that's named apatite. Apatite, or biological apatite, that we find in bone, in teeth, in all mineralized tissues we have, are different of stoichiometric hydroxyl apatite. Because in biological apatite, we can have cationic and anionic substitutions. And in my group, we have studied the substitution of calcium by strontium. So I know that the most part of you are dentists. I will show only that strontium is just below calcium in the periodic table. I think the last time we saw this, it was in the high school, right? You know, I came here for a PhD defense, and I asked to the candidate, no, where calcium is in the periodic table? And the girl said, oh, Professor, sorry, I'm a dentist. So I decided to come here and to show you that's where you remember about this. I'm joking, but because calcium and strontium share the same family in the periodic table of elements, they have similar chemistry. When we drink like mineral water, if you look in the ingredients, you see that we intake strontium daily in their diet. And due to the similarity with calcium, strontium attached mainly in our hard tissues. But what is important in this kind of substitution? That strontium can have a dual effect in controlling bond cells, the cells responsible for bond formation and for bond resorption. And we can take advantage of this effect. Since strontium enhances the activity of osteoblasts and controls the activity of osteoclasts, we can use strontium to treat bone disease like osteoporosis. In the past, strontium granulate was used to treat osteoporosis, and it was in the market, it was used in the clinics, in the form of this drug named Protos. What's the problem? This drug was discontinued. If you will look here, the dose of Protos administered to a patient per day was 2 grams. So can you imagine if you go and you intake daily 2 grams of any salt? You have good effects, like controlling bone formation and resorption, but we will have also side effects. And the side effect here was that strontium granulate was inducing cardiovascular mineralization. So chronic kidney disease and another kind of pathological mineralization. So this drug was discontinued, but we keep in mind that strontium has positive effects. In this way, we can try to study some substitutes for strontium granulate. This is here in the top, the structure of strontium ranelate. So now I got inspiration from nature to extract these flavonoids, or the flavonols, from flowers, from fruits, from vegetables, and we can use these binding sites of flavonols to bind the strontium. And now I can deliver strontium systemically using a natural compound that is antioxidant, anti-inflammatory, and compare the effect of this natural compound with the effect of strontium ranelate. We have used two kinds of flavonols, maurine and quercetin. I will show you now only the results with maurine, but we have also another study published with quercetin. So we used this kind of flavonol that's named maurine. We complexed this with strontium. We treated the cells, osteoblasts, primary osteoblasts, with strontium ranelate, strontium maurine, and only with the flavonol maurine. We found that using the same concentration of the drug, using the same concentration of strontium ranelate, we got higher alkaline phosphatase activity for the flavonol maurine complex. And if we look here in low concentration, strontium ranelate had no effect compared to the control that were the cells cultured in the absence of these compounds. We also got higher mineralization when we used strontium maurine compared to the control mineralization by the cells. And the explanation we found for the enhanced mineralization was the enhanced expression of the gene related to the formation of alkaline phosphatase. So everything here was related to increase in the expression of alkaline phosphatase, the increased activity of alkaline phosphatase, thus increased the mineralization by osteoblasts. But we also investigated the effect of maurine and strontium maurine on the differentiation of osteoclasts. And we see here that compared to strontium ranelate, that SCRR here, strontium ranelate, using the new compounds, we completely reduce the differentiation of osteoclasts using low concentrations. So if the problem was the concentration of strontium ranelate that was dangerous, we found a good substitute for this drug that can control the differentiation of osteoclasts, at the same time enhancing the activity of osteoblasts. It is interesting here for the students that were working with flavonols, that you showed in the morning, that the effect of the flavonol itself was similar in the control for the control of osteoclast differentiation compared to strontium maurine. So the flavonol itself has an effect. The difference here was the effect of the complexation in the osteoblasts activity. I can try also to imitate the structure of mineralized collagen fibers using the synthesis in confined medium of calcium phosphate nanotubes. So here we used track-etched polycarbonate membrane, and we precipitate minerals inside the pores of these membranes. The pores of the membrane had the same diameter of the mineralized collagen fibers. And when we used the confined medium, the template, to try to imitate what's happening in vivo, we got this kind of fibrillar mineralized structure. And in the absence of confinement, we got this kind of amorphous structures. I think it was disconfigured. I had some TM EJAX images. Oh, no. Yeah, I had here, oh, sorry. In the top, I had the TM images, but it's not appearing here. But we show also that this nanotube, we added strontium to the structure of the nanotubes, and we see here in yellow that strontium was distributed homogeneous through the structure of the nanotubes, and we had no phase segregation. So we also prepared here material substituting calcium by strontium. These nanotubes were bioactive. I mean, after the immersion in a simulated body fluid, we precipitated selectively a hydroxyapatite at the surface, exclusively at the surface of the materials. These materials were able to deliver locally strontium, and depending on the concentration of strontium we had in the nanotubes, and more importantly, in therapeutic doses to induce physiological mineralization and not pathological mineralization. I think it is also configured here, but I think maybe, I don't know. I said to you that I have no animation, something like this, but that's okay. The point here is we also studied the effect of these nanotubes in their resorption by osteoclasts, and the structures completely reduced the ability of osteoclasts to resorb the mineralized area of a hydroxyapatite plate. So another kind of material that we can apply for controlling osteoporosis and also to implanting osteoporotic models. In the absence, controlling the precipitation of apatite in the bench, in the laboratory, we are able, even in the absence of the confined medium, to get the formation of this artificial apatite with a structure very similar to bone apatite, and not only in the morphology, but also in the structure, as we can see here by comparing the X-ray diffraction of the bones and the apatite that we precipitated. We see that all the peaks are very similar and match the peaks of the X-ray diffraction of the bone. And also the infrared spectrum of the particles are very similar to the infrared spectra we found for the bones. I will show you the result that we had with this kind of particles to treat dental hypersensitivity. You know that strontium is in the form of strontium acetate is the active ingredient that we find in sensogen rapid relief. So more than used for the control of osteoporosis, strontium has been also used to treat dental hypersensitivity. Then we used these kind of particles, and we applied these particles at the surface of dentin. And if you will see here, the particles were able to deposit at the surface of dentin. In special, the particles containing strontium coated much better than the surface compared to hydroxyapatite. Okay, but if in sensogen rapid relief we already have strontium, what we are doing to put with another kind of strontium? We should realize that strontium acetate is a soluble form of strontium. So if you guys here have used sensogen rapid relief, as soon as you stop brushing the teeth, 2 days after, you have the hypersensitivity again. So it's not a persistent treatment. So the idea here was to use a compound with lower solubility and with a higher affinity by the surface of dentin. And why not trying to imitate the mineral structure that we already have in the dentin? So that's what we found here. We also used strontium carbonate nanoparticles to try to treat, to block dentin and twist the deposition at dentin surface. And we also treated human dental pulp stem cells with these particles, and we saw that the particles, the strontium carbonate particles, were able to enhance the differentiation of the stem cells in a mineralizing phenotype. Why to use now strontium carbonate, not phosphate? In the, you know much better than me that in some toothpaste, we have calcium carbonate as an abrasive agent. So here we're trying to get the biological effect of strontium with the abrasive effect of carbonate. And we're trying to do some abrasion, also remineralizing in this simultaneously. And more recently, we prepared, because we were trying to produce this in a product, in a commercial, in the form of a commercial product. So we put the strontium apatite particles in a gel composed by type 1 collagen that is similar to the protein we have already in the teeth structure. And we compared the effect of this treatment with gels of polyvinyl alcohol containing the nanoparticles. So the hypothesis here was, okay, if I have collagen, I will have a better adhesion at the surface. And that's exactly what we got. Here, I show you the treatment of eroded dentin. In this case, the dentin were eroded using EDTA. And here, we treated the eroded dentin with collagen and hydroxyapatite containing strontium. And here, the same particles, but we used not mimetic gel. And if we see here, the gels with collagen were much more efficient in coating dentin comparing to the PVA gel. These particles were also able to induce differentiation of the stem cells in a mineralizing phenotype. Another way to deliver these particles is incorporating them in bond cements. So we prepared here some PMMA bond cements. We added the nanoparticles, and we implanted these bond cements containing the strontium phosphate nanoparticles in the femurs of rabbits, the tibias of rabbits. So the structure of these bond cements, here it's better, shares similarities with the structure of trabecular bone. The micro-CT analysis showed that we have a porous and interconnected structure that can also favor vascularization and cell penetration. The materials were able to stimulate mineralization by osteoblasts. The cells were able to penetrate the structure of the cements. The materials were able to release strontium and calcium. So all the effects, the positive effects we found in the regulation of osteoblasts and osteoclasts were related to the release of these ions by the materials. And after implantation, the presence of implants induced a higher expression of Osterix-related gene that is a late marker of cell differentiation. Then, as a last example, I will show you some materials that we are preparing, containing collagen and a polysaccharide that is able to imitate, to mimic the structure of glycosaminoglycans in presenting the bond extracellular matrix, like chondroitin sulfate. If you guys or you in your family had some problems with cartilage formation, for sure the orthopedist said to you to intake type 2 collagen or chondroitin sulfate. But I have to say that systemically it has no effect. I'm not going in depth in this discussion, but locally, we can have good effects in bone and cartilage repair using these two compounds. But the problem with collagen and these glycosaminoglycans, like chondroitin sulfate, hyaluronic acids, is that they have very poor rheological properties. So in the preparation of scaffolds, membranes, and hydrogels, we have to increase a concentration, so the materials get more expensive. Sometimes we increase the concentration, we get precipitation, not the formation of organized structures. So we're trying to replace chondroitin sulfate in the fabrication of biomembranes with collagen by this polysaccharide that's named carrageenan. Carrageenan is a sulfated polysaccharide, and the structure of carrageenan is very similar to chondroitin sulfate. And carrageenans have much more suitable rheological properties for the preparation of biomaterials. Carrageenans are isolated from renewable sources, so we have lots of advantages of using this kind of material. Then, so we prepared this collagen kappa carrageenan scaffolds, and we cultivated osteoblasts at the surface of the materials. The scaffolds contain only collagen and collagen carrageenan. We found increased mineralization here and increased alkaline phosphatase activity for the osteoblasts in the presence of the scaffolds, in special the scaffolds containing kappa carrageenan. In the presence of carrageenan here, called plus 5% kappa, we also increased the expression of the main genes related to the differentiation of pre-osteoblasts in a mineralizing phenotype. So alkaline phosphatase, osteocalcin, osteopontin, bone cell protein. So our hypothesis that was the substitution of, or the addition of material similar to a glycosaminoglycan could enhance the performance of osteoblasts was confirmed. Then, just to finish, as a final remark, I hope I showed to you that we can replicate the mineral and the organic part of the bonds in vitro. We can take advantage of this replication, not morphologically, but to prepare functional materials. Strontium can promote mineralization, activating alkaline phosphatase gene expression, and reducing the differentiation of osteoclasts. The use of strontium is versatile due to the similarity with calcium, but we have also to investigate possible side effects when we are using strontium. And we have studied this in the group. I'm not showing here because of the time, but we have really showed that increased collagen and strontium concentrations has bad effects on the structure of hydroxyapatite, and also it disturbs the organization of collagen. And increased concentration of strontium in the materials can also reduce the attachment of osteoblasts on the materials. And the organic matrix can be mainly replicated by using phospholipid-based films and biopolymers like collagen, polysaccharides, and another kinds of materials, always to try to buy the functional, new functional biomaterials. So I'd like to thank you again for the invitation to be here. I have here the main collaborators that directly participate of the results that I showed you, and the students from the group. Here is the more recent photos, but I'd like to thank all all the students that have contributed to these studies. And thank you for your attention.
Thank you, Anna, for your wonderful presentation. Any questions?
Congratulations, Paula, for your very great presentation. I don't know if I understood that compartment that you deposited the strontium, right? So it's for delivery of those strontium, right?
Mm-hmm.
Or, yeah, okay. It's to be in the surface of a material, like in plants, or because you create the nanotubes and then we deposit the strontium into the nanotube, right?
No, we precipitate strontium together with already in the structure of the particles that we are depositing. So I replace calcium by strontium, like I prepare the precursor solution containing calcium chloride, strontium chloride. I precipitate hydroxyapatite in the presence of both the ions.
Okay.
So I have strontium and calcium incorporating the structure of the nanotube.
Okay, I got it.
So it's completely
Because I thought you deposit afterward. Then I'm gonna ask you if there is any implication of the size and of the pore and the length of the nanotube.
No, it's precipitated in a homogeneous way,
Okay,
in situ and together.
Okay, thank you.
Any more questions? So thank you, Anna, again, for accepting all my invitations. Obrigada. So for finishing the activities for today, I would like to call Professor Manuela and Professor Renata to come here with me, please. I also would like to thank the Post-Graduation program of Oral Biology and Oral Rehabilitation, and thank you for helping me to do this. You are so supportive and important as well. And I would like to thank you, everybody who helped us to organize this event. And to finish, I would like to thank again the professors Valentin, Owen, and Ana Paula to come, and all of you for always accepting my invitation. And say again, as I said in the morning, all of you are so special to me. Thank you again for coming. A gente fala para o Bruno encerrar? É Bruno? Vitor. Vitor, você está aí? Vitor foi com Deus. Acho que eu vou... vamos tirar uma foto com os palestrantes?
Are you hungry? Pessoal, a gente tem um copo break, deu muita energia, então...